of all, I want to thank Professor Murase for inviting me here to Kyoto, to this conference.
Can you use the microphone?
Oh, yes, thanks. Okay. I always forget these things. I'm going to start my stopwatch, too, so if I get over in time, just start throwing things at me, because I know it's almost lunchtime, or it is lunchtime. So, yeah, so it's conferences like this, at least for me, that you get all these people from diverse backgrounds under one theme, and I see, like, for me, I get a lot of really good ideas and new perspectives. And this topic in particular, this topic of what is evolution, lies close to the sorts of things that I'm really interested in thinking a lot about. So, as an evolutionary biologist, I've come to see that there are lots of themes that keep jumping out again and again. And one in particular, one thing that seems to keep jumping out and out, and it's related to the sorts of things I'm going to talk about today, is the difficulty in detangling the ecology from the evolution from the ecology. In order to, the one good thing about that is we can actually take advantage of this property as evolutionary ecologists and do all sorts of kind of cool things that I'm going to show you. But the thing is, it requires fast-evolving systems. So the things that I'm going to talk about today have been done in two different systems: E. coli, a model system for these sorts of things, and then also an artificial life program called VITA. And if you haven't heard of it, I'll describe it throughout the presentation. But I'm going to actually focus on the VITA portion of this study. So, talking about themes that exist within evolutionary biology, another theme that exists is this profound tension between random processes and deterministic processes. Professor Murase mentioned it, kind of talked about a little bit of these sorts of things in his introductory talk the first day yesterday. You have natural selection that's working to adapt populations to their environments, but this selection requires random mutations. So it's this intertwinement of random processes and deterministic processes that because of this, you get this contingency on the history of an evolving population. And there was a sort of movement that was going on in the probably late 1970s, early 1980s, where evolutionary biologists were starting to recognize the importance of historical contingency. And probably the most outspoken proponent of this idea is Stephen Jay Gould. Of course, like Lewinson and many others have been talking about the importance of history, a historical perspective. But Stephen Jay Gould was one that really came out strong with it in his book, Wonderful Life. Pretty famous book. But what's really pointing, at least to the argument I kind of want to make, is that in this book, during this sort of trying to get across this idea of historical contingency, he came up with this thought experiment that's used all the time in evolutionary biology. It's this idea of replaying the tape of life. If you could jump back to some point in history, sometime in the distant past, if you could start life again, how would the world be different? So, I mean, these little circles just kind of represent different worlds, groups of organisms or environments. In Stephen Jay Gould's case, any replay of the tape would lead to evolution down a pathway radically different from the road actually taken. So, in other words, if you ran it again and again and again, you'd get totally different worlds. But there are those evolutionary biologists, and in this case, Simon Conway Morris is an example. It turns out he's actually a friend and colleague of Stephen Jay Gould, and Gould speaks very well of him, but Morris disagrees with Gould. He thinks that the evolutionary routes are many, but the destination is limited. I really like that particular quote. And if the people on Stephen Jay Gould's kind of camp are right, then evolution, contingency renders evolution fundamentally unpredictable. So, as an evolutionary biologist, I think one of the coolest experiments I would do, if I could give any experiment, would to go back, because I think a lot of us would like to go and tell us a lot about the history of life. I mean, I think just that, but obviously it's not a very easy experiment. However, we're lucky because we've got a lot of talented biologists that have thought of ways of replaying this tape of life. And so the most famous example of replaying, at least I call it a tape of life, it's a small section of life, is Lenski's long-term evolutionary experiment. It's going on right now. It was actually started in 1988. Lenski actually originally started working on insects, and he's kind of one of these impatient scientists. He wants to see results. I kind of consider myself one of these too. He wanted to see results. He wanted to rerun this tape of life. So what he did is he started he started an experiment using microorganisms because of their tractability and their fast reproduction. And he started in 1988. He just took an isogenic strain of E. coli, and there was actually one gene that was different. It was a marker gene between there was six strains and then 6 of another, and he seeded this in a minimal glucose solution. So this is something that glucose eat. And what he does every day, and he's been doing this for over 20 years. He's got people helping him, of course. But he goes in and at the beginning of the day and takes out 10% of the solution. And he puts that in another flask of the same sort of minimal glucose solution. And then he lets it grow all day and usually gets 6 to 7 generations. And then he does it again, and he does it again, and he does it again, and he does it again. And now in 2009, it's still going, and now we have over 45,000 generations of this particular strain. But the cool thing that I like to always emphasize about this experiment too, I've had a chance to go work a little bit, or we've done some visits at Michigan State, and I've seen the whole process, and it's so simple, yet that's what makes it so elegant, I think, is that he freezes about every 500 generations. So this is the beauty of this particular experiment, is that if you freeze these every 500 generations and you find something cool later on, you can actually go back and replay it again and see if it happens again. Or, here's the coolest part, you can actually compete the current organisms to their ancestors and see how things have changed over time. So this is what he's done. He's taken, I keep doing this close. In 1980, he seeded it. Around 2,000 generations, he started to see some properties. So who's right, Morris or Gould? He wanted to know this sort of thing. And what he's found so far is they're kind of both right. That's the thing, is that's probably the most interesting part of it, is that they're kind of both right. One, he's found general properties that exist in all 12 lines. One, for example, the property that they all kind of gotten a little bit bigger and more efficient at metabolizing glucose. That's a general property that exists. There's other things, like there are several strains that have the supercoiling of the DNA. There are differences, though. There are certain strains that actually have mutator genes in them that kind of they're more prone to replication errors. And there's all sorts of things. If I had time, I'd go through all the cool things that have come about. But the thing that I want to tell you, if you've not heard of this, I think it's one of the coolest things in evolutionary biology. I actually told my grandma this, and she thought it was super cool. But it's this idea that at about 31,000 generations, there was a strain, one of the 12 strains, developed the ability to metabolize citrate. Now, for those who don't know much about ecology biology, it turns out that that's one of the defining features of E. coli, is that it can't metabolize citrate. Evolutionary process. The first question is kind of probably the most obvious question. How do we get that? How did this E. coli strain get to a point where it was able to utilize a sugar it's never been able to utilize before? What were the mutations that it took to get to that point? The second question relates to the replaying of life. How repeatable is this particular event? Could, if he went back in time, he is actually working on this sort of thing, and he's working on all the genetics, they're doing all sorts of interesting things with this. How repeatable is this particular event? An event this dramatic. But the question that I'm interested in, most relevant to this particular talk, is this question. Could you actually decrease the time? Could you decrease the time and speed up the rate of adaptation? That's what I'm really kind of interested in the project that I'm doing. That's the question that's kind of the central question of this little project that we've kind of started and had some fun doing and working on. Can we speed up the rate of adaptation? That's a question a lot of people have had, but it's easier asked than done. So first of all, you need a system that's suitable, and I'll get to that's one of the reasons we use Avita. But before I get into that, I know most people here are comfortable with fitness landscapes, so I'm not going to go into the detail. But fitness landscapes have been a really popular metaphor with evolutionary biologists ever since the introduction by Wright in 1932. We use them all the time. A lot of times we misuse them. But probably the defining features are the z-axis. The height of these landscapes represents fitness, which is kind of opposite the way that physicists think about them. Physicists kind of flip these, well, we as biologists flipped them, I think, before, because physicists were using them, I think, before. And then the x and y axis kind of are abstract versions of genotype space. But the main thing is that selection drives populations uphill. So you can have a point, but in this case, you can think of it as a cloud point. as natural selection, it pushes things up here. So under strong selection, there will be times, assuming a multi-peaked landscape, where presumably populations will get stuck at suboptimal peaks. So if this is the ancestor, well, this isn't working. Well, that doesn't work. Okay, so if this is the ancestor strain, and this is its landscape, selection pushes it up. But if there's strong selection, there's going to be times where you could get caught in suboptimal peaks. And so one of the questions that biologists talk about all the time, and actually Dr. Iwasa brought this up in his tunneling. He talked about tunneling, the same sort of thing. How do you kind of jump over or get through these valleys? So that's kind of the question that we're dealing with here. And what are the mechanisms by which that can happen? Because you imagine it probably does, because things do tend to move over a given enough time. So here's the question. Here's what we're interested in. Here's one strategy. There's lots of strategies, but this is the strategy, the first strategy that we're starting to attempt to do. Let's pretend you're stuck on a suboptimal peak up here. And what you do is simply just change the environment after it's stuck. That's the strategy, is change your environment. So you have environment A, it gets stuck. So there are lots of ways of moving, but here's the idea that we're kind of playing with. If you just simply change the environment, if you change the environment where you place it in a different part of another landscape, because these landscapes are defined by the environment in which they're in, could you then generate movement of this population again? And if there's movement, and then you switch it back to the original environment, is that a way of tunneling, so to speak, to a higher point? And so that's kind of the strategy. Now, like I said, there's other strategies to talk about, but that's kind of what we're talking about. So here's the question that gets at the original questions: can we speed the rate of evolution? How do populations evolve in changing environments? If we can start to understand this, and it happens like we're kind of predicting, or we have, I mean, this isn't even, a lot of people have been talking about this, but it's actually quite understudied, that this is the sort of question we want to get at, is how do populations evolve in changing environments? So, like I said before, to do this sort of study, it requires a system that's well understood, that's tractable, and that has a fast generation turnover rate. So that's why we've kind of turned to digital life as one form. Now, E. coli is one form, just like Rich Lenski uses, and then also in one of the labs I work on with Ben Kerr, he also uses E. coli for long-term evolutionary experiments. But I'm not going to really get into the details of this, because first of all, I'm not an expert in AI. This is a new field for me, and we kind of refer to our collaborators, Charles Ofria, who's kind of the originator, him and Adami and Brown from Caltech, that came up with the Vita. But just to kind of give you just a brief, like, this isn't really a history, but there are different forms that we can use to study this question. But we've chosen to use a Vita for lots of reasons. It was one of the first organisms, well, it's claimed in the literature that where you get complex organisms from simple replicators. Now, something like amoeba is kind of cool because it's, I think, the first example of the emergence of self-replicators from non-replicators. But anyway, we don't really need to go into that. I just want to say that we're using Avita, and there is a reason why we use Avita. The main reason we use Avita is because it was really the first AI program. Because the idea of using AI to look at evolutionary questions has been around for some time. But the reason why Avita is, I think, a little bit more special than the other ones is it's been adopted by a really well-known biologist, and it's been published like crazy. So we didn't want to really reinvent the wheel. We're still learning Avita. It's very complicated. I don't know all the guts of it. I'm learning every day about it. But I mean, these are some of the publications it goes. I mean, it's certainly, I think, been somewhat adapted by at least some parts of the scientific community. But the main idea about the virtual world of Avita is that it's a computer program that contains these evolving organisms called Avidians. They compete for CPU time. And the way that they're rewarded is if they can compute specific logic functions, then they will be given a reward. So let's say there's a logic function and. If they can do that, if it's an environment that rewards and or equals or in these other logic functions defined by us, then they'll be rewarded and they'll replicate faster. So that's kind of the main idea. And they're represented by these like strings of sequential primitive computer instructions that you don't really need to know the details. If you want to know the details, I'd be glad to try to explain them afterwards, but I'm still trying to learn the program myself. But why Avita? The main thing is this very thing. So we don't, I mean, people know more about it than we do. So this is kind of a small divergence, since the title of the conference is What is Evolution? I think Avita and AI is always a fun area to talk about really what is life and what is evolution. But there's been several people that have come up with really what they think the ingredients of evolution are, and that's simply replication, variation, and differential fitness. Now, some people would change that list a little bit depending on what sort of systems they're talking about, but these are, I think a lot of people can agree, these are kind of the ingredients for what is evolution. Sort of, or at least what's required to see evolution. And I think the people that have started Avita make a pretty darn good argument that Avita displays these sorts of properties. So the other thing that's kind of cool is using Avita-like programs and being able to compare it to our carbon-based life that we know. Here's an example of potentially life that isn't I guess, stymied by any of the sort of historical accidents that potentially the biochemistry that we know of has, and it's a way of comparing. So John Maynard Smith had this nice quote on why it would be a waste of time, I guess, to look at these sorts of things. So I'm going to show you a quick little prediction of the sorts of experiments we're going to do, and I'm going to show you some results, and then we'll be closing in. So first of all, there are two regimes that we have. So if we want to compare the effects of changing environments, we need two regimes. So one regime is the static regime, where you simply put it in an environment. In this case, it rewards this task. And you can change these tasks to whatever. There's lots of tasks. But you can put it on, and equals. And you run it, and you see what happens to the fitness. Now, if we go into the dynamic environment, now we want to see how a changing environment affects the overall fitness or the rate of adaptation. So in this case, gray is the exact same environment. Whatever we define gray as, we'll say. And blue here is any alternate environment. Now, that's one of the areas we're investigating. There's all sorts of different ways to do this alternate environment. You can do orthogonal environments. You can do like overlapping functions where there's similarity, but there's just a little bit of difference. You can alternate these environments at a particular frequency. We want to keep it simple. For the first set of experiments, we just wanted to go from the reference environment, the alternate environment, and back to the reference environment, and compare that to the control. If there's an increase in fitness, then maybe there's suggestive evidence. So this is what you would expect if you just ran it in a constant environment, and here's the dynamic environment. Here's where you get this, you change the tasks here, and here is this environment in this section. So now I'm going to show you kind of what our prediction was before. So this is a prediction. I'm not saying this is necessarily what our results were. This is a prediction where at this point, because everything is measured relative to the reference environment, what you get is you would expect a drop because it's in a different environment. It's evolved to really a set of functions, the ability to do certain things, and so you put it in another environment, you expect it to drop. And then there shouldn't be really any total, there'll be maybe neutral changes, but nothing that will really improve the fitness in that alternate environment. What we hope is if you're hopping around on that landscape is that when you get to the original environment, you're in some other area of the landscape, and then you're able to maybe potentially go higher in fitness, or depending on whether you're just talking about overall fitness or rate of evolution, we want to see what happens. So here's some results. So this is one example of a Davidian over time. These updates are simply, it's approximately the amount of time it takes to execute 30 instructions. You can define these updates or time in these AI worlds in different ways, and the fitness is relative to the environment that you're looking at. So in this case, same sort of reward functions that I showed in the previous slide. What you see, these circles represent times in which there were changes in the genome. Now, I've marked on here where certain functions, the ability to perform certain functions occur. So mutations, all of these are mutational steps along here. Just like walking on this landscape is walking around via small mutational steps or big mutational steps. These are mutational steps. That's why we represent it like this. It can be in any different way. But what you see is, this is usually the easiest function. Those that know a lot probably know more than I do about the logical functions. Not is one of the easier ones to evolve first. If you give it a set of instructions in random set of numbers, this is the one that will evolve first. And then over time, something else can pop up. These plus minus just means sometimes it will lose. It'll have a mutation where it'll lose the function, but generally it gains it back really quickly, and then over time you will get this increase. So that is one. So 5 minutes? Okay. So that was in a static regime. This is a dynamic regime. So this is a situation where you've run this particular the same sort of run, but at this point you switch it, you give it to the other environment, and then you see what happens to fitness. Fitness is significantly higher if you compare it to the previous one, and I can show there's issues about comparisons of similar organisms, but I just want to show you what in one Vita run, what it looks like over this dynamic regime. The blue line here represents the fitness in the alternate environment. We just put that in there kind of for fun. But really, we're always kind of looking at the reference environment. So some of our predictions were right. We're seeing some surprising things in Vita, just like we do with E. coli. But the thing is, this is the sort of pattern. So we've done thousands of these, and we want to see if we can get statistical significance, of course. So we do this over many, many, many runs. And we basically we're replaying the tape of life, at least in Vita, hundreds and hundreds of times. And it takes, well, actually, even though it's a computer program, it burns a lot of CPU time. It takes a lot of time to do this. So the colors aren't showing up very well, but what you're looking at is 407 replays of time. And using the static regime, which is this one right here, and the dynamic regime. I really like this way of presenting the standard error. The fuzz around the line, the mean is the dark line, the fuzz around it is the standard error, which shows you throughout time what statistical significance is. What I'm showing too here, you can show all sorts of things, is the dominant organism at the end of the run. Then we retrace its history. There's all sorts of different ways of doing that. So that's kind of what you're looking at there. Now there are other groups that actually have results that are also that support the same idea. Recently, Cashman et al. In PNAS recently published something in 2007. A little different perspective, looked at modeling networks and the structure of tRNA, and looked at whether changing environments increased the speed at which it predicted these structures. And they found the same thing. These are for different populations. They get different. They show different. They do show speed, but the other difference is they go at a higher frequency at some places. So that's kind of cool to actually look and see that other studies also are getting to the same sort of results. One of the other areas is, again, changing that environment in a particular way. One way is to do it orthogonally, like I said, so that just means reward functions that aren't in the reference environment. So if you have not and equals, you don't have those in the orthogonal. So another way of doing it is getting into a topic that we've talked about a little bit, constructionism, or niche construction. And this is kind of cool. It's kind of, you can think of it like this. This is not a really great metaphor, but it's kind of like walking on that landscape as if it was a waterbed. I don't know if you have waterbeds, but there are these, as you move, you kind of affect the landscape a little bit. Or it's kind of like being on a teeter-totter, where there's a fulcrum, and as you move, you actually affect the landscape around you. Basically, it's rewarding on a frequency-dependent manner. So if you're doing a function that nothing else can do, or that no one else is doing, you get rewarded higher proportionally. If everyone is doing that same function, even if it's rewarded in that environment, too bad, you don't get as high as a reward. And there's a lot of evidence within natural systems that niche construction is an important form of a selection regimes like this. So, basically, I just kind of want to summarize here with, again, or just kind of reiterate what the strategy is and what the underlying theme is. The idea is, we'd love to replay the tape of life. Unfortunately, it's quite impossible to do that. We could, if we had the ability to look at all topologies and all environments and all genetic combinations, we could run these, we could figure out the same sort of thing, but we can't do that. So we kind of want to take a top-down approach at this sort of problem, and we want to see, are there algorithms or strategies for increasing the rate of evolution? And why is that important? It gets to kind of the last question of my slide, which could be maybe a conference on itself later on. But it gets to something that I think is a really exciting area of evolution biology that's growing and growing. And it's this right here. How can we apply evolutionary principles to other realms and other substrates? So this is the question I think is quite exciting. And you can apply maybe these same strategies that people figure out, some of these work, into areas of computer science, optimization, engineering, robotics, maybe even art and architecture. So anyway, before I end, I wanted to tell you who the real players of this are. Charles Free is the starter of Vita. Ben's done helped with a lot of coding. Ben is at the University of Washington and kind of spearheaded this. Josh is kind of the lead on this project, so I definitely want to give credit to them. And here's some of the funding that we have. But thank you for the time. Let's go have lunch. Yeah.